We're starting into a new part of the course where we're covering the life cycle of the cell. From here on out, we're going to really be looking at how chromosomes are arranged, the genes on the chromosomes, and how they assort into potential offspring. Ultimately, what we're going to get to is trying to figure out where our traits are going and how they're getting there. So the first part of this is talking about the cell cycle, how the cell grows, how the cell reproduces. We're going to specifically talk right now about the parts of the chromosome. So it's just a chromosome map, looking at the different pieces and how those, to give us a shared vocabulary. I'm hoping that by the end of this, we know what a centromere is, what sister chromatids are, and what a kinetochore is. That's the goal. Life moves in cycles. I'm not sure if you guys remember way at the beginning of class, we talked about frogs. I gave you this big pseudo-mental experiment about how to design a frog, or not how to design a frog. They're already designed, but the three-legged frog. And back then, it was great. You guys didn't talk ever. Now, you do. Welcome to knowing each other. It's a little bit more fun now, I think. The first few weeks of class are always killer. It's just like, get to know you. Who are you people? Do you know me? But we talked about frogs. We talked about tadpoles becoming froglets, those froglets becoming adult frogs, and the adult frogs laying eggs to become more tadpoles. It's a cycle. Birth, life, death, birth, life, death, over and over and over again. Cells are no different. Omnicellulae cellulae. That's Latin. It means all cells come from cells, all cells from other cells. This was proposed by Rudolf Virchow. It's a part of the unified cell theory that we talked about way back when we discussed the parts of the cell. It all sort of comes back. All cells come from cells before them. Now, that again goes to the idea that maybe there was something before the cells. For about 4 billion years, we've got evidence of cells. 4 billion years, every cell has come from a cell before it. Every cell has come from some other cell. That begs the question, what came first? You know, chicken or the egg type thing. If you're interested in early cellular evolution, how the first cells arrived without other cells present, take 102. I do a whole lecture on the chemical selection involved in the first cells. But for this course, every cell comes from a cell before it. There might be some slight changes, and those changes can accumulate over time, but they all are relatively the same. This cellular division, where you're dividing cells up, you're taking one cell and making 2 daughter cells. Those daughter cells have more daughter cells, and so on. Is very, very, very highly regulated. The timing has to be precise. You need to make sure that things are happening in the right sequence. There are going to be times where it's right to reproduce and times when it's not right to reproduce. Some of you guys have made that decision already for yourselves, right? When now is not right, or it might be, I don't know, you live your life. But there is a right time in your head. For me, I definitely did not have, my daughter's going to hear this one day, I did not have my kid at the right time. I needed to respond to my environment. I did not have any resources when I had my first kid. Literally, I had no insurance. That sucked. $25,000 child. I'm keeping a tab of it. She'll repay me later. I didn't have the resources I needed. Cells, though, have to look around them. If they have resources available to them, they can reproduce. If they don't reproduce. They stop themselves. They check things out to make sure it's safe. If you're living in an environment that's not safe, like a war-torn country, probably not the right time to have a kid. If the cell is under attack, it's not going to reproduce. It's checking out its environment. Let's see, if you have the resources. Oh, and if you're big enough, I know that sounds bad, if you're mature enough, you don't want to have kids when you're 10 or 11 or 12. Biologically, the human body may be capable of it, but it's not ideal. Similarly, you don't want to have kids in your 60s. It's possible, but man, that would suck for the last years of your life, wouldn't it? Trying to raise a teenager when you're like 74, 75? Yeah, I know. That's the plus side of having my kid when I was younger. I'm so looking forward to retirement. I'm going to travel. I'm going to do things without that one. It'll be great. The cell has to precisely time its reproduction. How does the cell check out what's in its environment? How does the cell know what's around it? It sends out probes, all right. What's it probing with? What do you think it probes with? What do you think it's using to detect the environment around it? Okay, think about this. How do you detect the environment around you? Your eyes, all right. Touching things, all right. What do you touch things with? Your hands, and your hands have the skin on them. What's the equivalent? What? The membrane, right? The membrane has all these proteins in it that's checking out its environment all the time. It acts like its skin and its eyes and its ears and its nose, constantly sensing the environment. The extracellular matrix even, the exterior of the membrane. You got all of these things checking out the environment to see what's going on, to make sure it's fine on the outside, and also you've got some internal mechanisms on the inside making sure you're ready to divide. So in order to divide the cell, we have to have these structures that help us move the instruction manual of the cell around. Now, what are the instructions of the cell held on? What language of the cells is the cell's manual? Let me put it a different way, because I think I just confused you. We've talked about the different parts of the cell, and we talked specifically about the nucleus, right? What's housed in the nucleus? DNA. That DNA is the instruction manual for the cell. It tells the cell how to build itself. Not only how to build itself, but how to build the next generation. Every single cell in your body has DNA in it, and it's identical DNA. The cells in your body, except for your sex cells, they're different. But the other cells in your body have DNA, and you have to organize that DNA. DNA is organized into chromosomes. So all chromosomes are bits of DNA wound up really tightly around proteins. The instruction manual bound into a booklet form. You got lots of different pieces here. Here I've highlighted a few of the parts of the chromosome. I doubt I'm going to have you draw a chromosome map, but it gives you a visualization of it. Typically, you see chromosomes drawn as an X. What that is you've got two sister chromatids off to the sides here. You got one on the left, you got one on the right. The chromatids are identical to each other. Where they come together and join is called the centromere. It's the center. Wrapped around the centromere is a kinetochore. It's like a belt of proteins. This is what you can hook other things to. It's if you've seen, or maybe you, but some construction worker out wearing a tool belt, that's sort of like what the kinetochore is. It's a belt that allows you to put extra attachments on. And the tips of the chromosomes are called telomeres. So a chromosome is, in a certain phase, is made up of chromatids bound together at a centromere, surrounded by a kinetochore. For most of the cell's life, it's going to have its DNA really, really open weave, because this is an instruction manual, right? All of the pages of the book it's literally strewing all the pages around you so you can look at it and see the whole manual. You can take any piece of it and read it. Only during mitosis, the division of the nucleus, do you condense the chromosomes to make them easier to move. So this is sort of like open weave. Imagine a bowl of pasta, cooked pasta. Bowl of cooked pasta, pretty big. You can pick it up. You can touch any strand of pasta in there. Sorry, I'm hungry. Chromosomes would be like uncooked pasta in a box. Which takes up more space, a bowl of a pound of pasta or a pound of pasta in a box? The bowl takes up a lot more space, right? I always underestimate that. The other way I thought is that I prepared. Check this out. I went into the bathroom. It really freaked people out too. And I stole toilet paper. So this toilet paper is tightly bound up. Like, you guys have been in a restroom and you've run out of toilet paper at some point in your life, and you've probably screamed, like, not screamed, but you're like, give me toilet paper, bring it to me. That's what children are for. They're good at it. But toilet paper rolls are really tightly compacted. You've seen all the advertisements for them. They're like, this roll has like 50% more toilet paper. You can't really tell when you look at it. Nobody goes through and counts the sheets. Nah, it's just thinking. That'd be really funny to count the sheets and be like, write the company. Be like, you shorted me 3 sheets. They might send you a coupon. Lifetime supply of toilet paper. So the idea is this is really easy to transport. Here, your hands are out. Catch. Look, see? That easy. I can move it. Okay, throw it back over here. Look, we can move it all over the place. Super simple to move. This is a chromosome. It's tightly bound up. The problem is I can't read it all. If the instructions are written on this, I can only read the outside. I can't read it that's in the middle right now. Does that make sense? These are instructions on how to build proteins, and I can only see that little bit. How do I see the ones in the middle? You open it up, you unravel it, right? So I can unravel this. Taking up a lot more space now too, right? It's not compacted, it's open. I'll just wear that. So now I can get to any piece and read the instruction manual. Do you see what I'm going with this? This is chromatin versus a chromosome. Man, this stuff's everywhere. Okay, there. All right, so that's a single chromosome. But the deal is, in eukaryotic cells, you don't have just one chromosome that encodes for your set of instructions. In fact, you have 2 of them. It's like you have 2 copies of the manual, 2 copies of the instructions on how to build you. 2 chromosomes that have the same genetic coding regions, the same instructions, are going to be called homologs. So here you have this red one, here you have this blue one. These are going to encode for about the same genes, but they may have slight differences, very small differences. So homologous chromosomes are very, very, very similar to each other, but they could be different. This is not to be confused with the chromatids. Here you have a red chromatid on the left and a red chromatid on the right. These red chromatids are absolutely identical. But that blue chromosome and the red one are slightly different, so they're termed homologs. Just slight differences. In you and us, I keep saying you. I'm one too. We're eukaryotic. It's like I'm this alien being that's come down to teach you biology. We eukaryotic. Now it seems forced. Now it's like he really is an alien. Or an amoeba in disguise. Us eukaryotic chromosome. Okay, restart. Eukaryotic chromosomes that are in all of us, all right, good, are inherited in pairs. You get one of these chromosomes from your mom, you get one of the chromosomes for your dad. Your mom and dad are slightly different from each other. You got to admit that. I mean, if they were the exact same, it would be really weird. They have slight differences, and that can be accounted for on their chromosomes. Well, you get one from mom and you get one from dad, and that makes you. So now you're a totally unique individual. These homologous pairs are not absolutely identical, but they do encode for the same basic traits. They encode for the same instructions. They evolved from the same chromosome. So here you might have moms, here you might have dads. They're pretty darn close to each other. So here's the important slide, and the one that I always end up really glossing over. On these chromosomes are genes. So remember, chromosomes are strands of DNA. A piece of DNA that encodes for a protein or a polypeptide is called a gene. So a gene is a polypeptide encoding region. In fact, I'm going to ask you guys to write that down verbatim just so we're all on the same page. A gene is a polypeptide encoding region of DNA. So any gene, a polypeptide encoding region found on one of a homologous pair, is at the same place on the other one. Here's one gene. Here's the same gene on the other chromosome. So if you look in that box up there, the gene on the left is going to be a homolog to the gene on the right. They encode for the same basic thing. Let's give you an example. The ability to taste, what is it? Phenothiocarbamide? Okay, here's the deal. How many of you guys like broccoli? A few of you, a lot of you, okay? How many of you guys like Brussels sprouts? Great. So here's the deal. There is a particular chemical in Brussels sprouts and broccoli that is very bitter. In order to taste that chemical, you need to have a certain protein that your body makes on your tongue, on the cells of your tongue. So far, so good? If your body is making it, let's say that's this blue one here, you have the protein present, so you can taste bitter. Taste. Is broccoli good to you? Brussels sprouts? No. If you're tasting something bitter, it's not good. It kind of sucks. You know, little kids are like, this is terrible, and you're like, no, it's not that bad. Turns out, genetically, it could actually be bad. It's interesting because we don't actually truly understand what how each other experience food. It's different from the way okay, this is going to blow your mind. It's almost like seen in the Matrix. The way you taste steak may be very different from the taste that I taste when I eat steak. Genetically, when somebody who has this PTC gene, who has the ability to taste PTC, eats Brussels sprouts, they want to spit it out because it is so bitter. Some people, though, may have this other version of the gene at the same place. That version of the gene is broken. It doesn't make any of this protein, which means if you're not making the protein, you cannot taste the bitter flavor. All you're tasting is the sugar in the Brussels sprouts and broccoli, which is sweet. Totally different taste. That's what is going to happen with these genes. When you have two different versions of genes, they're called alleles. So alleles are different versions of the same gene. We're going to get into much more depth on those when we get into Mendelian genetics. But I wanted to introduce the terms because they come up a lot. So, to refresh what's going on in this slide, which apparently is way more in depth than it looks. On a homologous pair, you'll have the same genes at the same locus, which means location. If those genes are different on one chromosome than another, you're encoding for the same protein product, but there may be slight differences in instruction that lead to alleles. One may work and one may not work. I'll try. Okay. That's okay. I'll do it from the beginning. You've got a homologous pair. On the homologous pair, you're going to have the same genes at the same locus on both sets of chromosomes. Where those two genes are different at the same locus, you formed alleles. Alleles are alternate versions of a gene. So you get, of your homologous pairs, you get one from mom, you get one from dad. Basic sexual reproduction. You're going to produce these See, I almost slipped into science talk. You're going to produce sex cells in your body, sperm and egg, respectively, male and female. Each of those is going to carry 1/2 of a full set of chromosomes to you. So when they fuse, you get a whole set. Now, homologous pairs are very, very, very similar to each other. These homologs are different by less than 1% gene sequence. Truthfully, we're not terribly different from each other. We may think we look different. You know, we got different heights, got different hair color, different eye color, little things. And I really do mean little things. Because the vast majority of our bodies work exactly the same way. The same neuroreceptors in my brain are just like yours. The way my intestine digests things is pretty much the same as yours. Does that make sense? Internally, we're pretty much the same. And it's because we have so much homology. So many of our genes are so similar to each other. In fact, we're only about 3% different from chimpanzees. I mean, that's not far at all. I think we're almost 60% related to bananas. Yeah, it's about 60% bananas. Why? We're eukaryotes, bananas are eukaryotes. And all of those bits and pieces of the cell that you need to make a plant cell are very, very similar to what we need to make an animal cell. Now, there is an exception to this. The sex chromosomes, you could have an X chromosome and a Y chromosome. They are very different sizes. The X chromosome is a robust chromosome full of all sorts of wonderful genes that do an enormous amount for your body. The Y chromosome is a little piece of leftover genetic crap. It's got like 3 genes on it, and it's got a lot of virus DNA. That's about it. It's a placeholder. But that X chromosome, that's important. So those are different, but all the rest are the other homologous pairs are very, very similar to each other.
Cells dividing is probably one of the most important processes that our body can regulate. And it is extraordinarily highly replicated. You have to be able to respond to the environment. You have to be able to get everything right every single time and consistently. You've got billions of cells in your body, each of which have had to at least multiply once. If there were any mistakes, they need to be accounted for and fixed, or that cell needs to die. In this mini lecture, we talked about chromosomes as long strands of DNA wrapped around histones. So you've got long strands of DNA that get really condensed around histones. As it divides, it looks like the chromosomes shorten and thicken, but they're just hypercoiling. And that's just there to make it so they're easier to move back and forth around the cell. We talked about the structure of the chromosome, where 2 chromosomes contain the same genetic coding regions, and they're super similar to each other. They're termed homologs. You get 1 homolog from mom, you get 1 homolog from dad. Eukaryotic chromosomes, like you, are inherited in pairs. You've got a pair or 2 of each unique chromosome. Your mother gives you 1, your dad gives you 1. So those pairs of chromosomes come together to form the eukaryotic cell. On a chromosome, you'll have genes. Genes are polypeptide or protein encoding regions. They might be found in one homolog, and then that same gene will be found in the other homolog, even if there are slight differences between them. So we went over chromosomes, long strands of DNA wrapped around histones that seem to shorten during cell division in order to allow them to more easily be transmitted to daughter cells. These content review questions will help focus your studies. The next lecture we're going into is going to sort of dive in depth on chromosome sets. So in this lecture, we talked about individual chromosomes. In the next lecture, we'll talk about sets of chromosomes and how we can interpret them.